The dendrites and cell body of a neuron may receive thousands of synaptic connections from other neurons. When these neurons are activated, they release neurotransmitters that open ligand-gated ion channels in the membrane of the receiving neuron. As ions flow through these channels, the membrane potential of the receiving neuron changes at the point of stimulus and passively spreads along the membrane. Some of these synapses shown here in green are considered excitatory because they make it more likely that the receiving neuron will fire an action potential. Excitatory signals depolarize the membrane, making the membrane potential less negative. This change is called an excitatory postsynaptic potential. Inhibitory signals, shown here coming from the red connections, hyperpolarize the membrane, making the membrane potential more negative and making the neuron less likely to fire an action potential. This change in the membrane potential is called an inhibitory postsynaptic potential. If the neuron receives enough excitatory stimulation, the membrane potential will depolarize sufficiently for the receiving neuron to fire an action potential, beginning in a region called the axon hillock. The changes in membrane potential of the axon hillock can be measured with a voltmeter. The voltmeter measures the difference between a recording electrode inside the cell and a reference electrode outside the cell. This value is normally about -70 millivolts, which is the resting potential of the neuron. If enough excitatory signals arrive close enough together in time, they may depolarize the membrane above a certain voltage, called the threshold potential. At this point, an action potential is generated. The action potential propagates the signal along the entire length of the axon. A look at the voltage-gated channels in the axon reveals how action potentials self-propagate down the axon. At the resting potential, the inside of the cell is negative relative to the outside of the cell. When a depolarizing potential reaches the axon hillock, where the voltage-gated channels begin in the axon, the inside of the cell in that region becomes less negative. The change in voltage triggers a shape change in the voltage-gated channels that causes the channels to open. Sodium channels are the first to open, and sodium ions enter the cell. The influx of positive charges further depolarizes the cell membrane and results in a positive moving spike in the membrane potential. The voltage-gated potassium channels open soon after the sodium channels and begin to release potassium ions outside the cell. As these positively charged potassium ions continue to leave the cell, the sodium channels begin to close, and the charge across the membrane in this area returns to its previous state. The voltmeter reading dips back toward the resting potential. A delay in the closing of the potassium channels causes a brief undershoot of the resting potential. The depolarized region is returned to normal as the sodium-potassium pump shuttles ions across the membrane, reestablishing the resting potential. Meanwhile, the changes in the membrane potential have opened adjacent sodium channels, and more sodium ions begin to enter farther down the membrane. The process continues in one direction, down the length of the axon. If this neuron makes an excitatory synapse with the next neuron, it too may fire an action potential.